chemists in my group, it's a mass spectrometry group. We spend a lot of time developing methodologies working in the metabolomic epidemiology space and the exposome space. Like many of us here, we're working in this idea of this population-based phenotyping paradigm, where we can perform stratified medicine, we can take a group of individuals, stratify them by some magical molecular markers into discrete subgroups of unique therapeutic trajectories, unique treatment regimens. This is a wonderful concept, but of course, how do we identify our magical molecular markers? For us, we focus on urine. My lab works primarily in pulmonary diseases, and so I spend a lot of time at lung conferences explaining why I look at the urine if I'm looking at a lung disease. Most MDs working in lungs realize that physiologically, the urine is quite far from the lung. So, why urine? A couple good reasons is that, first and foremost, it's non-invasive. We're working with pediatric studies or doing time course or interventions. It's very easy to collect from that standpoint. We're very really volume limited in urine. It's happening. It does happen, but generally not. More importantly, again, speaking from the lung field, we find that the signals in blood are actually amplified in the urine. Particularly if we look at direct challenges in the lung, we can see molecular signatures in the urine with much stronger effect sizes than we can observe in the blood. And lastly, of course, similar to blood, it's an integrated metabolic signal. And so we find it very, very useful. Of course, there's a very long history of working with urine, and we're in good company over thousands of years of using the urine to influence information on the physiological state of the individual. But as a chemist, there's a lot of challenges in working with urine. What you're looking at here is sample urine from a cohort of asthmatics we'll talk about later today. And you can see we have a heterogeneous distribution of matrix composition here. As a chemist, this is what happens. And so we took urine samples and we spiked them with an isotopically labeled internal standard. And we just did a standard dilute and shoot approach to this. You can see the RSD here is huge. And this is just an internal standard, same concentration in all samples. So 38% RSD is just due to the matrix heterogeneity. If we're trying to detect subtle changes in metabolites or exogenous compounds, well, that type of RSD, good luck. So what we found is that we need to control matrix effects. And so, for example, if we dilute with the specific gravity you see here, prior to analysis, we can get RSDs down to a level that's much more reasonable. The key here is you have to do this prior to the analysis. Almost everybody corrects urinary data by some sort of urinary output metric, creatinine, specific gravity, etc. But we do it prior to the acquisition. There's a lot of different ways to do this. I won't go into all the different methods out there. As I said, we're using specific gravity, which is shown here. This is a handheld refractometer. And it works very well, but it's exactly that. It's a handheld refractometer. So if you want to do this at thousands of samples or tens of thousands of samples, measuring them one by one on this takes a lot of time, and it's certainly not fit for purpose for what we want to do in metabolomic epidemiology. So what we did is we decided that, wait, we can use a refractive index detector as a way to calculate the specific gravity. And that can be automated. So we have it in 96 well plates, it's an autosampler, automate the whole process to calculate the specific gravity. This is the Blood-Altman plot showing the limits of agreement between the two different methods, the handheld refractometer and the RD detector. You can see the limits are very good. This has been confirmed in over 800 samples. So we had a method that could enable us to measure specific gravity, to do it rapidly, to automate it, and be part of our global pipeline. I don't have time to talk about this today, but it's been published. You can go to the publication, happy to talk about it later in the talk. What's important about this is that it is fully automated. So if the samples are sent to us in our requested tubes, we never actually touch the material. So the tubes go directly to the robot. Shown here, we use robotics to aliquot it to daughter plates. We measure specific gravity. We normalize every sample to the same specific gravity value. We extract it, seal the plates, and get it over to the mass spec. All fully automated, never touching the sample. One thing that's so fantastic about this is now this system can be run, we have undergraduates who are doing all the work. It's so robust that under supervision, this can be successfully run by undergraduates. And the proof is in the data. So here's some raw data. This is pre-QC correction from 24 batches, so 24 96 well plates of metabolomics data. We use what I call technical internal standards, shown here just to monitor signal performance over time. And so this is the RS or the CVs of our technical standards This is pre-QC correction, this is post-QC correction. You can see that we can get our CVs and the QCs less than 5%, and in the samples less than 15%. This is for 24 batches of 96 well plates. You can get these types of CVs. And this is where we want to be if we're able to detect subtle metabolic shifts. So what we're trying to do with this, the idea is that we can take an individual, look at their metabolic profile, their metabotype. And in my case, if we look at lung disease, we want to be able to use the metabotype shown down here to inform or identify treatable endotypes. So instead of coming from the top, which is what the clinic does now, is we look at the symptoms, the clinical symptoms or the phenotypes of the individual to identify subtypes of disease. We want to go from the bottom up and look at their metabotypes so we can have a quantitative metric to stratify these individuals. A study that we've been working on for quite some time is shown here. It's called UBioPred. It's the unbiased biomarkers in the prediction of respiratory disease outcome. So it's one of the larger studies that has been done looking at molecular phenotyping of severe asthma. It's one of these large European things where they put extreme amounts of money. We had 85 different partners in this. It was incredible. Going into very briefly the study design. We have 4 cohorts: severe asthmatic non-smokers, severe asthmatic ex-smokers, mild to moderate asthmatics, and healthy volunteers. We had at baseline roughly 600 individuals, and the severe asthmatics were also resampled 12 to 18 months later, another 300 individuals. So roughly 900 individuals. At the time when this was collected, this was already back in 2010 when the study was instigated, this was a huge study. Like today's standards, it's relatively small, but back then, this was a very big deal. We have a huge amount of patient metadata, and we've essentially put every omics in the book we've thrown into this study. So it's a fantastic study for transomics or interomics work, and I don't have time to talk about that today, but there's some really amazing things happening with this. We're focusing only on the urinary metabolomics. What we do here is we take the metabolomics data, and this is just hierarchical clustering. Then we just put it into what we call a polar dendrogram here. You can see we have 7 different clusters of biochemical information in relationship to asthma severity. And everything in red is significant in either the univariate or the multivariate line. So looking at this in more detail, here are the 7 different clusters, and you can see that some of them go down with asthma severity, some of them go up with asthma severity. There's unique biochemical information in there on a compound-specific basis. What's a little more interesting, this is a canonical variant analysis. And so here we can look at the 4 different cohorts. And so what we see is that our severe asthmatic non-smokers are distinct from our mild to moderate asthmatics, but they overlap with other controls. Now, one of the issues you're dealing with in metabolomics is, generally we're looking at people who are not well. They are sick, they have a range of morbidities or comorbidities, and most of them are beyond medication. In this case, the severe asthmatics are generally taking oral corticosteroids. From a metabolism standpoint, we know that this has a major effect on metabolism. So if we're taking high-dose OCS, we know this will have a metabolic phenotype. So we need to be able to delineate that and any findings that we want to talk about. So what we've done here is we stratified individuals. Here are severe asthmatic non-smokers who are taking OCS, and here are the ones who are not taking OCS. And that's very important for looking at analysis. Here's the loadings plot shown here. And what's key for the talk today, we're going to talk about, is that the main metabolic signatures driving this separation in severe asthmatics are these carnitines over here. Lots of carnitines. So what we did is we took the carnitines and we z-scored and concatenated them to make a composite carnitine score. And as you can see at the group level, you can see that carnitines decrease with that severity. Now we had a lot of other omics data available from this cohort, so we started to mine that. We looked at the transcriptomics data, and we could see that using some enrichment score analysis, our fatty acid metabolism also decreased with asthma severity. And then we looked at the carnitine transporter itself, and we could see that the transporter, the SLC22A5, the mRNA levels also decreased in the sputum. So we started to see there's a systemic dysregulation in carnitine metabolism associated with asthma severity. So this suggests that this could be a potential intervention for severe asthma. But our question is that not all individuals are going to have the same type of asthma. We're looking at this as a heterogeneous syndrome. We want to be able to identify subgroups. We wouldn't propose that necessarily this would be appropriate for everyone. We need to look at this with more granularity. So we moved to another study, which I don't have time to talk about in a lot of detail, but this is called RAS, or Refractive Asthma Stratification Program. And this is a UK cohort that's designed to look at the utility of a biomarker-based approach to treating asthmatics. And so based on specific asthma biomarkers, can they stratify people for specific therapeutic regimens? It's a very interesting study. But again, for the purposes of today, we're looking at the urinary metabolomics. So we applied the same methodology to this. We did the same types of dendrograms here with the clustering, and now we have 6 different metabolic clusters here. But then what we've done here is we've divided the individuals based upon their T2 status, so T2 inflammation. We have T2 high individuals, and we have T2 low individuals. And now we can see how the metabolites, how they shift between T2 high and T2 low. In some clusters we see a difference, and in some clusters we don't see a difference. Doing the same type of PCA analysis, we can see that we do see overall shift in the carnitine levels, or sorry, the overall metabolite levels between the 2 different groups, T2 high and T2 low. This is the loading spot, and what's driving this very quickly on the cluster basis is, again, it's the carnitines. And laser pointer does not really work, but there's the carnitines. So we make the same type of composite carnitine score, but now it's divided up by T2 high and T2 low. So now we can see that, more importantly, is that these findings, they align with the earlier work in carnitines, but that we wouldn't specify that carnitines would be beneficial for everyone. It depends upon your T2 status. So the individuals who have a T2 low type of severe asthma would be the ones more likely to benefit from carnitine therapy. This dysregulation in carnitine, it further supports what's being developed in the asthma field, that at least part of asthma severity is related to mitochondrial dysfunction. And it suggests that we need to include carnitine levels as part of a panel for doing molecular phenotype approaches in asthma. But what people are more interested in here today, I assume, is trying to understand how we want to put this in the context of life exposures. And so working with a colleague at the Karolinska, Dr. Eric Ulen, showed up here. He's a pediatrician working in asthma. We've been putting a lot of thought into this space, and he and his colleagues published this a couple of years ago in Lancet Respiratory Medicine, where they call it their GET-omics approach, where GET is gene, environment, and time. So using omics over time to look at the gene-environment interaction. So how is the life course exposures related to the development, in this case, of asthma and COPD. To look at this, we have a cohort called BAMSA. This is a fairly well-known Swedish cohort. Again, I won't go into a lot of detail, but here's the cohort dynamics. I imagine many people in the room have heard of this over the years. Has one of the best acronyms. BAMSA is sort of like the Swedish version of Winnie the Pooh or something. This is a very, very well-known, beloved cartoon character for children. And it stands for, in Swedish, B is children, allergy, environment, Stockholm, and epidemiology. So it's a very nice acronym. We have extensive information from these individuals, and we have multiple biosamples over 20 years that they've been monitored. So we were lucky enough to get the urine from these individuals and look at the metabolomics at 4 years of age in the same individuals at 24 years of age. And so based upon retention in the cohort, we had 578 pairs. So we have the same children, or same individuals at 4 years and 24 years of age. So we decided to look initially at the associations between air pollution exposure and the levels of the urine metabolite. Now, before I show this, I should give a caveat that this is brand new data. I wanted to present something new and novel today, and so I got these results on Thursday. So it's not a polished story yet, but I thought it would be more interesting to get something sort of hot off the press versus something that's already published. So this is the study population for the metabolomics work to draw your attention. So here are the air pollution levels that we determined. So we looked at PM10, 2.5, and NOx. So this is the 4-year-olds and the 24-year-olds. To explain what we're doing here is that we looked at levels in the first year, so from birth 1 year out, and then the year prior to biosample. So for the 4-year-olds, we looked at air pollution levels at year 3, and for the 24-year-olds, we looked at air pollution at 23. And we looked at those associations. Looking at the air pollution levels, you can see, so here from the 4-year to 24-year-old, you can see that actually the levels are decreasing. So this tells us, particularly for NOx, we see decreases. So overall air quality in Stockholm is improving. So over 20 years, we do see a reduction in the air pollution. So in terms of what we observed, we applied the same method I've been talking about today to this, following QC and curation of the data. From the untargeted metabolomics, we had almost 10,000 features, right? All these metabolomic features from both negative and positive ionization. But from our targeted work, we had 260 chemical standards that were, or compounds that were identified with chemical standards, so a lot of confidence in these compounds. So what we see here, this is just a summary, and it's only so much I could do from Thursday, but we see that there's an association, a significant association between air pollution exposure and the overall level of the urinary metabolome. So exposure to PM10, PM2.5, and NOx overall increases the urinary level of metabolome. The full change you can see here is very modest, but the p-values are quite decent. So we are seeing a significant physiological response in relationship to air pollution exposure in Stockholm. In terms of looking at across the life courses, we don't see a big difference between the 4-year-olds and 24-year-olds. This seems fairly constant, which makes sense in association with the air pollution exposures that they're experiencing. In terms of focusing on specific pathways, and this is one that came up predominantly in our work, this is the caffeine pathway. So caffeine itself is not dysregulated, the metabolites of caffeine are. And so we can see here, this is for PM10. This is the first year, and this is the year prior. See more or less the same thing. So a number of these metabolites of caffeine are all increased in relationship to air pollution exposure, particularly PM10. This is something we've seen quite a lot in literature. If you go and you look at the metabolomics data in relationship to air pollution exposure, dysregulation in these metabolites is something that seems quite consistent. We looked at the untargeted data as well, and we're happy to see that caffeine metabolism came out at the top, so same findings as our targeted data, that's reassuring. And then looking down here for the 24-year-olds, we see all of these in yellow, these fatty acid metabolism pathways. Those of you who know my work, my lab focuses on lipid metabolism. We're very interested in fatty acids. So we're super excited to see this. We particularly look at eicosanoids and lipid mediators. So when leukotriene metabolism came out, I was very excited. I thought, wow, that's great. So the PhD student, I said, show me your leukotrienes that are dysregulated with air pollution. I'm really excited to see this. So he gave me this table. Here are all the metabolites that came out in his analysis. So you can see we have a whole range of metabolites here. And what I can say is that they don't exist. As someone who's spent 20 years working on leukotrienes, those leukotrienes do not exist. If you go to HMDB, there is an entry for them, but they've never been reported, they've never been observed. So this is one of the problems with non-targeted data analysis, is that people get their annotated hits from a database, and they take it as true, and then it goes to a table, and it goes into a publication. I'm fairly confident that if I hadn't looked into this in detail, this would have wound up into the student's publication. He doesn't know this at all. None of the other co-authors are familiar with this. And I've seen it in a number of other publications out there in association with air pollution exposure, dysregulation in fatty acid metabolism, and eicosanoids. So this is a real issue, I think, in the field. What we can say is that overall, that Stockholm is a fairly clean city in terms of air pollution levels. But even then, we do see that there are metabolic effects associated with air pollution exposure. Even in this relatively clean city, we see a systemic upregulation in the urinary metabolome. Now, what are the health effects of that? I don't know. But there's been, of course, as this audience knows, many studies over the years that have shown a relationship between air pollution exposure and multiple health endpoints. So the fact that we're seeing this with early life exposures all throughout life, I think it's interesting, and it's something that we need to follow up on. And the data analysis has just started since Thursday, so watch this space. So where do I see this all going? In terms of speaking with an exposomics audience, I wanted to bring up some next steps. This is not comprehensive by any means. Some of the things I think we need to consider. The top one, I think, is the solid analytics. As a chemist, I can't speak strongly enough for the need for very solid methodologies to do this. I haven't gone too much into the details, but I feel this is really vital if you want to detect these often subtle changes. In addition, we need specific targeted methods in order to do this. And what I mean by this is that most of the untargeted work is done with database matching. And if you want to do that, then the metabolites you're looking at need to be in the database. And for a lot of these environmental chemicals of concern, we don't necessarily have appropriate metabolites. In my case, shall we say, urinary metabolites of chemicals of concern are not present in these databases. If they're not there, then how are we going to identify them with untargeted data approaches? And lastly, I think it'd be very interesting to be able to localize the in vivo exposure. So we look at the lungs, so to understand when we inhale environmental exposures, where do they spatially localize in the lung? What is the local physiological effect of the exposure? So give just very briefly in the time left what we're doing in this space. This is work that's being done at City University. So we're generating some of these metabolite libraries. And so here what we've done, this is for 32 fungicides were selected based upon their use patterns. Is that we take mice and via oral gavage, we dose them at extremely high levels of the fungicide. This is, first 25% of the LD50 and then 50% of the LD50. Urine is collected, and then it's run through the protocol we just talked about. So in terms of the specific gravity normalization, the same mass spec method, and then we collect all of the spectra to make suspect libraries of all these metabolites. So at the end, for 32 fungicides, we end up with 4,000 fungicide spectra we can then input into MS2 libraries. And so we can take these libraries and we can upload them to public databases. They're in Mass Bank, they're in MONA, so people can use these when they want to do their biomonitoring. For example, we looked at students from the City University, and in 73 of them, we found that this fungicide, tebuconazole, was detected in 2 of them. It allows us to start doing personalized monitoring of individuals. So you can start understanding, okay, these are the specific metabolites for specific exposures. We want to start screening for these, and we can start moving to personalized exposomics by being able to identify the exact chemicals of interest in individuals. But in order to do this, we need to have the reference spectra. They don't exist. So we either have to make them, or we have to predict them using in silico approaches. And so we're continuing this work, expanding this with additional environmental chemicals of concern, but it's not possible with all the chemicals. that would be a lot of mice. So we need to think of smarter ways to do this, but we can at least use some of these seed data to help train AI models to do sort of in silico predictions. So lastly, I think it's going to be really exciting to try and visualize the spatial exposures that are occurring here. So this is not my data, this is a paper that came out last year, but here they're actually looking at being able to visualize PFAS levels. This is in concrete, but there's no reason we can't do this in vivo. And so we're very lucky at the Karolinska. We work with thoracic oncology. Every week we get longitudinal resections. And so we take those back to the lab, and we can mount those. We do all sorts of ex vivo stimulations. And so we're currently doing work with different environmental chemicals of concern, particularly with microplastics and PFAS. We can expose these expanded lung tissues and look at the resulting physiological effects of these. And then using imaging approaches, this is mass spectrometry imaging, we can look at the actual presence of these and then the resulting physiological effect of the exposure. What you're seeing here, this is an anti-IgE stimulation in human lung. And we're looking at, in this case, these are eicosanoids, which are pro-inflammatory compounds. But we wanted the same type of thing with environmental chemicals. The paper for this was just submitted, so here's the QR code if you want to look at that in more detail. But I think being able to visualize these chemicals in space is going to be extremely exciting. And understand what are the local in situ physiological effects of environmental exposure. So to wrap it up, what do I see sort of metabolomics and exposomics version 2.0? As I said, I think the method development is really going to be key, but we need to have fast methods. You can't come and say, we need to do a 60-minute gradient, we need to do it in helic, reverse phase, positive, negative, we need to do GC. It's not going to be feasible if you want to do tens of thousands of compounds. So it needs to be fast, but it needs to be reproducible. And so, for example, for our 260 metabolites that we observed in the different studies I presented, we've now transferred that entire method to a triple quad. And so we have a targeted method, which is fast, cheap, easy to work with. We need to increase the certainty in metabolite annotation. And for this, we need to have the right MS2 libraries. So we need to find a way to generate suspect libraries for the metabolites of interest. Standardization is vital. I don't think we're going to get a metabolomics method. It'll never be fully standardized, but we need to have standards that we can use to allow us to compare our studies, so we can start doing meta-analyses and integration. There must be some benchmarking to be done. We need optimized precision, so we can have the ability to detect statistically meaningful small changes. We need to automate this as much as possible. As I said, I think the visualization of this is going to be extremely interesting, and this all needs to be done within an open data context. Without data sharing, it's going to go much, much slower. So with that, I think we can see that the promise of metabolomics is strong. There's a lot of potential there, but also there's a lot of work to be done. Thank you for your attention.